Good morning, viewer. Beyond the shore, here's a most amazing news for our viewers. Scientists at the Alio is to have discovered specific types of brain cells in mice that experience significant changes as they age. They also identified a distinct hotspot where many of these changes are concentrated, published on 1st January 2025 in Nature. These findings could lead to the development of therapies aimed at slowing or managing the brain's aging process. Key discoveries in aging brain cells. 1. Sensitive cell. Scientists discovered dozens of specific cell types, mostly glial cells known as brain support cells, that underwent significant gene expression changes with age. Those strongly affected included microglia and broader associated macrophages, oligodendrocytes, tenocytes, and ependymal cells too. Second, inflammation and neuron protection. In aging brains, genes associated with inflammation increased in activity while those related to neuronal structure and function decreased. Third, aging hotspot. Scientists discovered a specific hotspot combining both the decrease in neuronal function and the increase in inflammation in the hypothalamus. The most significant gene expression changes were found in cell types near the third ventricle of the hypothalamus, including tenocytes, ependymal cells, and also neurons known for their role in food intake, energy, homeostasis, metabolism, and how our bodies use nutrients. This points to a possible connections between diet, lifestyle factors, brain aging, and changes that can influence our susceptibility to age-related brain disorder. Our hypothesis is that those cell types are getting less efficient at integrating signals from our environment or from things that we are consuming, said Kelly Zinn, PhD, a scientist at the All Institute for Brain Science and lead author of the study. And that loss of efficiency somehow contributes to what we know as aging in the rest of our body. I think that's pretty amazing, and I think it is remarkable that we are able to find those very specific changes with the methods that we are using. To conduct the study funded by the National Institutes of Health, researchers used cutting-edge signal cell RNA sequencing and advanced brain mapping tools developed through NIH, the Brain Initiative, to map over 1.2 million brain cells from young 2 months old and aged 18 months old mice across 16 broad brain regions. The aged mice were what scientists consider to be the equivalent of a late middle-aged human. Mouse brains share similarities with human brains in terms of structure, function, genes, and cell types. Yeah, aging. Aging is the most important risk factor for Alzheimer's disease and many other devastating brain disorders. These results provide a highly detailed map for which brain cells may be most affected by aging, said Richard J. Hodges, MD, Director of NIH National Institute of Aging. This new map may fundamentally alter the way scientists think about how aging affects the brain and also provides a guide for developing new treatments for aging related human disease. Beyond this exclusive